Welcome everybody to the webinar on wastewater-based epidemiology, SARS-CoV-2 and beyond. I'm very happy and excited with this panel of experts who are going to speak, and we are going to learn a lot about this very exciting development that is happening in past couple of years during this pandemic. So before we go that, I just want to say that TIGS, the Tata Institute for Genetics and Society, is a institute, is a part of the Bangalore Life Science Cluster with the aim of using cutting-edge science and technology to solve problems of the society. We have three major programs. One is infectious diseases, another one is rare genetic diseases, and the third one is crop improvement using genome technologies. In infectious diseases, we have surveillance as one of the major vertical. And wastewater-based epidemiology of SARS-CoV-2 and beyond is something which is very close to our heart, as we think that this is going to be a major, we can make a major contribution to problems of infectious diseases, which India has a major share. I have had a privilege of being involved in this area from the very beginning of the pandemic. And in Hyderabad, we carried out wastewater-based surveillance, which gave a number which were very different from what was known at that time to be the level of infection. And it turns out that later, by the zero surveillance, that our estimates were accurate. In CCMB, Hyderabad and Gandhinagar team, you are going to hear from Dr. Joshi about it. These efforts have received the mention in WHO guidelines for the wastewater-based surveillance that has come out very recently. So this shows the importance of this work. In Hyderabad, in collaboration with many other institutions, we are doing some 20 open drain sites in several projects, and we are also, with the help of NIRI, Nagpur, we are covering several cities, not too very extensively, but to some degree, like Mumbai, Chennai, Nagpur, Delhi, Kolkata, and so on. Similarly, in Bangalore city has very well planned sewage system, maybe one of the best in the country. And with the support of BBMP here at TIGS, we are routinely monitoring 28 watersheds across the city. A precision health group on the other side is performing wastewater surveillance using drain sample from 46 drainage sites in Bangalore. So Bangalore team, where TIGS is playing a major role, is doing a great job, and it has tremendous contribution to of this study in the fight against COVID. And I think the Bangalore city model is going to be we can showcase for the rest of the cities in the country. The progress is really very well. As we have seen that COVID-19 pandemic has proved beyond any doubt that wastewater-based epidemiology can be used as an important tool to understand the disease prevalence in a community. And is not only the real scale of the infection, but also the variants that are contributing to it, which gives very important information of what dynamics is taking place and which part of the city may be doing better or worse, and based on that, the local authorities can take decisions. So it's extremely important approach. The advantages associated with the wastewater-based epidemiology is that it's very easy to do, is cost-effective. In fact, it costs nothing compared to an individual sample collection analysis. It's unbiased approach towards the public health system. So it is all really the crucial advantages, and of course, that is very, very affordable. And there has been tremendous progress in past couple of years in all aspects of this approach, which is sampling. So how to sample, especially in open drainage system, or even in the sewage treatment plants, what time to collect the samples and how many times in the week or day and so on. Then the molecular biology of the analysis, sample concentration, isolation, then amplification, and then the sequencing, and so on. So there has been tremendous progress, and equally or maybe even more important, the bioinformatic analysis and computation modeling. All these three aspects have seen great development in past several months. But at the same time, I think, as you will see from the speakers and following in discussions, that there a lot needs to be done, and it's so important that we cannot wait, and we must put best efforts to see that this technology is applied at a large scale, because the returns that it gives are tremendous. We need to make this more affordable, in the sense it can be applied to small places, especially less economically well-off countries. We have to develop means and system, or even I would say the culture of data handling and sharing for this, which is equally important. Otherwise, it becomes history. And the scalability and sustainability of this are really the challenges, for which there are lots of efforts going on. The government of India has floated several calls for projects to support this. And there are many other international organizations who are supporting a lot, and there's a big activity happening. And I'm sure this webinar is going to be very, very useful and educative for all of us. In today's webinar, as I said, on wastewater-based epidemiology of SARS-CoV-2 and beyond, we have two very prominent people of this in this area. Dr. Madhavi Joshi, who was one of the first one actually to carry on this kind of study, and not only contribute in the fight against pandemic, even publish so that methods become available to many others. And the other speaker is Dr. Aparna Keshvaya, and you will hear that why we think they are two very important speakers. So in the first, I'd like to invite Dr. Madhavi Joshi to make a presentation. And just a very brief introduction that Dr. Joshi is a joint scientist D and joint director at the Gandhinagar Biotechnology Research Center. She's among the first group of researchers who, as I said, started research in the area of wastewater-based epidemiology of SARS-CoV-2 in India. She has been actively publishing in several international journals, among which she has several papers which you all have seen. And instead of spending more time on that, she is very well recognized and for her expertise and contribution. I would request Dr. Joshi to give her talk, which is going to be on wastewater surveillance, a powerful tool for pandemic monitoring. So, Dr. Joshi, please can unmute yourself and start.
Yeah, very good evening, all of you. I hope I'm audible enough.
Yes.
And at the outset, I would like to thank Mishra sir and the entire team of TIGS for considering me to give a lecture on this wastewater-based epidemiology for the pandemic monitoring. And as sir mentioned, I would just briefly talk about GBRC. So we are an autonomous society under Department of Science and Technology, Government of Gujarat, and our focus is also addressing societal challenges using biotechnology. Our primary strength lies into genomics. So I still remember, when we had the first case of SARS-CoV-2 in India, in Kerala at that time, and then when government thought of imposing the lockdown, at that particular point of time, we were called by Honorable Chief Minister of Gujarat, and then they talked about what you can do as a biotechnologist, and we gave a series of lists that we could do genome sequencing, we could do RT-PCR diagnostics. Honestly, at that particular point of time, personally, I was not aware that the wastewater surveillance would emerge out to be a very powerful technique during the management of the pandemic, not only in Gujarat and India, but at a global level too. So today I will be presenting, a small piece of work. Although GBRC has worked in One Health aspect of the COVID-19, we started with genome sequencing from clinical samples, and then we also did studies on host pathogen interactions. We also did the detection of SARS-CoV-2 from non-human host. And then we also did whole genome sequencing from the wastewater and the non-human host and tried to understand the issue from the One Health point of view. But in today's talk, I would be only highlighting about what was the work that we carried out in the wastewater surveillance during the last 2 years of the pandemic. So we all know the background. Let us not go into the detail, but as on May 22nd, there are more than 522 million confirmed cases with over 6 million deaths. These are the WHO stats. And if we talk about the pathophysiology, even for the SARS-CoV-1, there were reports that the traces of RNA can be detected from the feces. And extending that hypothesis, these ACE2 receptors, which are also present in the small intestinal linings of humans, can also have replication of virus, which is shaded through the feces. And that is how, the potential of the wastewater-based epidemiology or the environmental surveillance of the SARS-CoV-2 is very important, because through the feces, this viruses or the RNA, it ends up into the sewage system, and which we are able to detect. There are many countries who have invested into the WBE. India, of course, now INSACOG is also extending its network for establishing the wastewater-based epidemiology network, who would be specifically looking at the major cities of India and trying to understand how the dynamics of SARS-CoV-2 is changing. Other than that, USA, Italy, Japan, Australia, they have also done some wonderful work during the COVID wastewater-based epidemiology. Now, what we should expect when we are investing into the wastewater-based epidemiology? So, of course, the qualitative detection of the disease, just the presence, absence, is also a very powerful thing, specifically in context of the individual facilities like, colleges, some institutions, nursing homes, or some vehicles like cruise ship and the flights, where we are trying to understand not screening the individuals, but just knowing whether the disease is absent or present. Then the second work, that is the independent quantitative estimation of the community level disease prevalence and the trends, where GBRC has focused more. So this could also act as our leading indicator, where we are trying to understand how the trend in a specific community is changing over a period of pandemic. What are the prediction potentials? What are its powers when we talk about correlating it with the disease trend? And the third thing is in the quantitative estimates of the rapid changes in disease incidences in case of certain outbreaks, or followed by certain large events or the festivals, how the community incidence is going to change. So these type of three major things that we could expect when we are talking about the wastewater-based epidemiology. So this is again, we have borrowed it from a WHO interim guidelines on the environmental surveillance of SARS-CoV-2. If we look at the bottom pyramid, which is primarily all the infected individuals, and then the second most important ladder in that pyramid is the environmental surveillance data, which could help us identify the infected persons who are shedding the virus irrespective of the symptoms or the hospitalizations. And then comes the importance of the diagnostic test, hospitalizations, and ICUs, and then the death. So you can see that even WHO has recognized that the environmental surveillance for diseases like this, of course, we all have success stories of polio, how we were able to catch the polio during using the environmental surveillance. But even in SARS-CoV-2, it has turned out to be a very powerful tool in understanding the epidemiology or the pandemic progress. This is again I have taken from the data produced in the WHO interim guidelines. So as you can see that, they have tried to give a holistic overview. So as per the WHO guidelines, the 100 copies per ml of the wastewater, they've considered it as a threshold. So beyond that, below that, any detection, they've considered it as a non-significant. So this is the limit of reliable quantification of SARS-CoV-2 RNA genome copies in the wastewater. As you can see here, the black graph, it represents the hospitalizations, and the different colors are the different variants, which we could say they are dominating different waves of the pandemic. And you could still clearly say that it gives us a very enough advanced time. So one of the major factor during the SARS-CoV-2 pandemic in a country like India was we were not aware about how the severity of the infection could be, what are the preparedness required. And in those type of circumstances, the wastewater-based epidemiology can give us a prediction early, so that at least we have our setups or our local authorities ready for the next pandemic curve whenever it is coming. So as Mishra sir also rightly pointed out, that there are very important considerations as far as the wastewater-based epidemiology is concerned. We are here talking about the data collection, analysis and interpretation. So what type of environmental sample? Either it is municipality sewage systems or STPs or the open drains or the surface waters, and then which method and the sample volume. So like automated composite sampling is required or the grab sampling is required. What should be the frequency? For example, we followed a frequency of a week, but where you require a full day frequency, what should be the ideal time of sample collection? So all these factors needs to be considered before implementing the WBE as a positive tool for predicting the pandemic. Then comes the virus concentration approach also. So there are many references. So although we followed a PEG based precipitation, but then there are other references where people have used membranes or the ultrafiltration techniques or two-phase separation techniques for concentrating the virus, because virus is very minuscule compared to the whole sewage, which is flooded with all type of flora and fauna, which is ending up into the sewage system. And then finally, the sample recovery and RT-PCR. So what are the QC checkpoints to ensure that you have recovered the RNA appropriately from the sewage systems? What should be, the internal controls, like some of the other enveloped viruses, and which methods of detection? All of us have now known that, during the Delta wave, the S-gene target failure was used as one of the major factor in diagnosing the possible clinical case of the Delta variant. But you could not do the same method in the sewage system. So identifying the optimum primers which would work and using the variety of primers to ensure that you have probably captured, all sorts of RNA through the concentrated sewage sample. So these are the key considerations. At the GVRC, I would like to present primarily the three type of the work that we have focused. So during the first and the second wave of the SARS-CoV-2, we were monitoring Ahmedabad and Gandhinagar primarily using the RT-qPCR technique. Then during the same time, we have also understood that how we could do genome sequencing using the samples which are collected from the sewage sites, and then understand how the variants are circulating with the help of genomics technology. And then later on, we have also optimized the same method in the digital PCR. So all these three things I will very briefly talk about. So this is the first example where we started the weekly surveillance of wastewater of SARS-CoV-2 in Ahmedabad, because Ahmedabad was the hotspot of the COVID-19 pandemic during the first wave, primarily due to the international airport and a heavy inflow. So in another independent study that we did with Oxford University of the genome sequences of clinical samples in the first wave, we could understand using phylodynamics that there were 100 different introductions of the virus in Gujarat during the first wave. Now, what here we did is that on the map, you could see that these are the different sewage treatment plants. So these were our sampling locations. The frequency was once in a week, and the duration of this sampling was September 2020 to February 2021. So this study was done in collaboration with IIT Gandhinagar, and this was initially funded for a limited period of 6 months by UNICEF. And in this work, so these are some of the representative images of the sample collection. At the same particular point of time, we used to collect samples from all different STPs and at a weekly frequency. This is the method that we followed for the virus concentration. So after collection of the influent and effluent samples, we first did centrifugation, and then the supernatant was collected, which was again filtered through 0.22 micron filters. And out of this filtrate, we took around 25 ml of the filtered supernatant and added the PEG for concentration, which was incubated overnight at 17 degree at 100 RPM. And then finally, the pallet down was done using cooling centrifuge, and then the final 300 microliter of the samples which was collected was taken for the RNA isolation using viral RNA nucleospin kit. And then it was a first wave, so we were doing the diagnosis for clinical samples also using TechPath. So we use the same kit for identifying the CT values. So I would just like to highlight over here, that if you look at September to February data, these are the genome concentration copies per liter of the sewage that we collected. We have collected some samples from Sabarmati River as well, just to keep them as control samples. And here you can see that we were clearly getting a higher concentration in the heat map during the third week of November, which was just, the effect of the Diwali festival at that particular point of time. In Gujarat, there was a major surge. So this paper, and then we also further analyzed this data with reference to percentage change in the gene concentration versus the percentage change in the confirmed cases of COVID-19, and we could get around 2 weeks of a lead time over here. And we have published this paper also in the Science of Total Environments Total, which has around more than 200 citations by now. So here, the first time we realized that the tool of predicting the pandemic using the wastewater-based epidemiology is going to be very, very relevant when, specifically being a government agency, when we have a very close interactions with policymakers and we are trying to tell them which area to be focused. So further, if you look at this graphs, in Ahmedabad, the north zone was the highest affected zone, followed by south and then west and central zone. As you can see here, this is an app data taken from Arogya Setu at that particular point of time, which also talks about concentration of the cases area-wise, and when we overlay our data of the WBE, there was a clear trend that we could identify some of the lead area which were responsible for surge in that particular cases. So these results we have also shared with the government organizations and the local authorities, and they have taken appropriate measures of increasing the surveillance on those particular areas. Other than that, because our key strength lies into genome sequencing, and we have sequenced by now more than 7,000 SARS-CoV-2 genome sequences. We also attempted genome sequencing of the SARS-CoV-2 from the environmental samples. So we had sequenced around 7 samples which were taken during September to February 2021 from Sabarmati and Vinzol STP. And those sequences are also submitted on GuySafe. So if you could just have look at, the uniformity of the SARS-CoV-2 genome. So it is from the river, it is around 62% uniformity. But in the February, when there was a peak going to be started of the Delta wave, we could achieve around 92% uniformity of the genome. And we have also analyzed these genomes for presence of certain key mutations like D61G, which are some of the potential mutations of the spike protein of the COVID. This is the sample which is collected from November in the Vinzol STP, and this is the sample which was collected in the 8th February. And again, we could observe some of the key mutations with the frequency of almost 98, 97, and 99%. One of the important things that I would like to highlight over here is that, in Gujarat, the Delta wave started somewhere from the 1st March onwards, very clearly. While these samples were collected on 8th February, although we sequenced it little late, but as you could see that this is a typical spike protein deletion in Delta that we have observed. And you could see the frequency of this mutation in the 8th February sample was already 57%. This was the second incidence, where we thought that these are a very important tools also in understanding the emerging mutations or, some frequencies which are present in those community SARS-CoV-2 RNA on a very high frequency. So this is another example. And this has also given us, an idea that how we should be addressing the future pandemic cases as far as Gujarat is concerned. So similarly, during the Omicron wave, in collaboration with the Veer Narmad South Gujarat University, they collected some samples from STPs in Surat, which is the second largest city in Gujarat. And we have done a whole genome sequencing with a uniformity of 85 to 90%, which is, very, very relevant, and also understanding the key mutations of Omicron, that is BA1 and BA2, circulating in Surat. The third thing that I wanted to highlight over here, and we all know that when we are attempting real-time qPCR, which is the mostly followed method for detecting environmental SARS-CoV-2 RNA. The digital PCR we also attempted because we know that this method works on the Poisson distribution, and the accuracy of the digital PCR increases when your target in the sample is in a very low concentration. So considering this, during the third wave of the pandemic monitoring in the Gandhinagar municipality area, we have focused these 5 STPs, again using the same method. Only the detection method changed over here. So we shifted instead of real-time PCR to digital PCR. And the detection was performed using the N1, N2 assay kit, which was recommended by CDC at that particular point of time. And we observed the similar cases, where if you look at the copies, genome copies in the wastewater against the confirmed cases, that we could get a lead period of around 14 days. So what I wanted to highlight very briefly over here, without taking much time, that when we are talking about COVID and beyond, which is, the theme of today's session, we could also use this technology of the wastewater-based epidemiology for understanding the responses and interventions finding outbreaks in the places which we think that they're already COVID free, or probably, the augmenting risk communication, because when we make this data public, like couple of countries have developed a dashboard of COVID-19 wastewater surveillance. So when people know that, their area has a higher copies of SARS-CoV-2 RNA, they will automatically take behaviors. Cost effective public health surveillance, I would say, because rather than testing 80-90,000 people from the green zone just to capture the rare events, the best thing would be to sample the wastewater or the STPs coming out from those regions, and then trying to understand the wave of the pandemic. This also gives us a very good leverage on the outbreak detection, because now, since there is we could say that there are no rising cases in India as a whole, except for a few outbreaks here and there. We could use these techniques also to understand where, the disease is going. Are there any changes coming up, or are there any places where we are going to observe the surges? Then we can also do the targeted surveillance, like Australia did a household or a society level surveillance, probably at least to the vulnerable or the high risk settings or the isolated communities, or maybe, the transport vessels like flights and cruises. And it can identify known variants, but I'm sure with more efforts, we could at least capture the mutation frequencies. If not novel variants, we could at least capture, the certain new mutation frequencies from the wastewater samples, and see whether they are connected with the clinical cases or outbreak or not. Of course, we are currently not majorly into biobanking, but this would be a very important way forward to have some of the samples stored, as to understand how this is moving forward. So, as Mishra sir said, that the work of GBRC and CCMB is also cited into this environmental surveillance of SARS-CoV-2 to complement public health surveillance interim guidelines of the WHO, released on the 14th April 2022. This is some of the media coverage of the work which is done in WB in Gujarat. We have almost 9 publications specifically focused on the wastewater-based epidemiology. And we have also come up with a total report which talks about contribution of GBRC in combating COVID-19 pandemic. This is a freely available document on GBRC website in case of those who are interested in knowing it further. So with this, I would stop share my presentation, and thank you all of you again for giving me the opportunity.
Thank you. Thank you, Dr. Joshi, for very detailed and very, very informative presentation. So we will take all the questions after both the presentations are complete. And I request all the those who are participating to put their questions in the Q&A part of the Zoom, so that we can address them after the presentations. So our next speaker is Dr. Aparna Keshwaya, who is a principal researcher at Mathematica. Aparna is an applied biostatistician with 2 decades of experience using advanced analytics and innovative data to clarify urgent public health questions. A nationally recognized expert in the field of wastewater-based epidemiology, she conducted seminal research that advanced wastewater surveillance for policy making and helped prepare communities to monitor the wastewater during COVID-19 pandemic. She currently works with the foundations and state and local public health agencies to develop better approaches to wastewater data analysis, synthesis, and reporting. Beyond wastewater work, Dr. Keshwaya directs projects at the intersection of population health and environmental health, which is so important now. It has always been important, but now we realize the importance of these. Before joining Mathematica, she led clinical trials in oncology and psychiatry that supported global diagnostic treatment decisions. Her research has been widely published in leading journals and it shows the significance of the contribution. Dr. Keshwaya holds a master's degree in biostatistics from the Harvard T.H. Chan School of Public Health. Is a 2006-7 Fulbright Fellow to India, and many of you will be amazed that she has a dual career as a Bharatanatyam dancer and choreographer. But today, you're going to hear from her about maximizing the value of wastewater data. So Dr. Keshwaya, please start your presentation.
Thank you. Can you hear me and see my screen?
Yes, perfect. Yeah.
All right, wonderful. So for those who may not be familiar, I wanted to just briefly introduce the company I work with, Mathematica. We are a policy research company with a mission to improve public health and well-being. And for more than 50 years, we've been mobilizing data, programming, modeling, and dynamic visualization to answer questions about health. We work with government agencies, foundations, and organizations in the US and internationally. In the field of wastewater surveillance, our work began in 2017 when we convened the first cross-disciplinary symposium in the US, and we brought together federal, state, local government officials with academic researchers and utility representatives to assess the value of wastewater data for the US opioid epidemic. 2 years ago, we pivoted to the COVID pandemic, and now we have national and international wastewater work underway. So wastewater is a relatively new data source, and I say relatively because when you look at the history of human tools and inventions, the first sewer system was actually developed 5,000 years ago. In 1939, during the US polio epidemic, a group of Yale doctors successfully isolated poliovirus in urban sewage samples. And since then, India, Israel, other countries have used wastewater-based epidemiology to measure the success of polio eradication efforts. Wastewater data have also proven valuable to study substance use. And that's because wastewater data can tell you not just whether there's any drug use in the recent past, which is what traditional survey data sources tell you, but also the types of drugs and the amounts of drugs being used in a community. So keeping in mind this multifaceted public health potential of wastewater monitoring, today I'll touch on 3 aspects of our research to maximize the value of wastewater data. First, I'll summarize some results from a national survey we conducted to characterize the role of wastewater data in COVID pandemic management in the US. Next, I'll discuss some work we did triangulating illicit opioid use through data synthesis in one US state. And then I'll wrap up by describing some international research that we have underway to assess equity in both national and global wastewater monitoring. So this first project I'll discuss was motivated by the fact that we are more than 2 years into the pandemic, and many public health officials are still struggling to determine the added value of wastewater data above and beyond clinical data. And so to hear directly from these officials, Mathematica has worked with the Rockefeller Foundation to jointly develop a 10 to 15 minute online survey. And I'll summarize some of our findings, which were also recently highlighted in an article by CNN. So the questionnaire included about 50 items, which covered 5 domains: factors influencing pandemic management, awareness, value, feasibility of wastewater monitoring, the current landscape of wastewater surveillance, barriers and supports to using wastewater data, and finally, future interest in wastewater surveillance. And after a 9-week fielding period from mid-November through mid-January, using a convenience sample, we received responses from 12 state public health agency leaders and 194 local public health agency leaders. And we had good coverage of rural and non-rural agencies and agencies with and without wastewater surveillance in place. When we looked at the factors influencing pandemic management, the traditional COVID data sources, case counts, deaths, ranked most influential. Environmental data were least influential, and on average, wastewater data was rated only moderately useful for pandemic management. Digging further, we noted that only 7% of local public health agencies without wastewater surveillance said they have what they need to begin. And the most common barrier to implementing wastewater monitoring was a lack of internal staff capacity. Remember, these are public health agency leaders in the midst of emergency response, and starting up one more thing was very difficult for them. More problematic for me as a biostatistician by training is the fact that some agencies said they didn't understand how to interpret the data or how to synthesize it with other sources. And so to help address this gap, we're in the process of developing an analytic tool based on patterns we see in wastewater data that officials can use to synthesize their local data with other COVID data sources. We will make this tool publicly available later this year. And the goal is for this tool to ingest structured wastewater data and then output a wastewater-informed risk score and alerts. In the survey, we also asked agencies that had implemented wastewater surveillance about the factors that supported their programs. And we heard that partnerships and government buy-in were the most cited supports to implementation. About half of the agencies with wastewater surveillance had used the wastewater data in pandemic management decision-making. And the top uses were to tailor communications to the public, to coordinate response with other agencies, and to target testing or vaccination efforts. Of the agencies that had not acted on their wastewater data, many said that decisions or actions were based largely or only on other data sources, or that the wastewater data were not timely or localized enough. And that's still a challenge that needs to be addressed in some areas. Looking to the future, most state agencies surveyed said that they are likely to monitor wastewater in the future, but only 21% of local agencies said the same. But another 40% said they felt neutrally about monitoring wastewater. And so this suggests that there's still room to move the needle and to make this innovative approach to surveillance more useful for public health officials. And that has been the focus of our work with the Rockefeller Foundation, to help public health officials easily act on wastewater data so that they can improve the health of their communities. Going forward, the officials surveyed were most interested in measuring new pathogenic viruses or bacteria in wastewater. The substance use also ranked highly in the US. And so that brings me to our work to triangulate illicit opioid use through data synthesis. To date, very few wastewater researchers have brought the wastewater data together with more traditional public health data sources. And to illustrate how this type of data triangulation can lead to very practical insights for policymakers, I wanted to showcase a comparative analysis that we conducted just before the pandemic began. By way of motivation, the US opioid epidemic has been impossible to control for many reasons, and one of which is a lack of timely and spatially granular data on changing patterns in drug use. Rural areas are particularly challenged by inadequate data. So for example, the US state of Montana, the map on the left shows opioid prescriptions by county, and you can see by the colors that there's a fair amount of information and variability. But not all prescriptions for opioids are problematic or illegal. And so if we instead focus on a more relevant outcome metric, which is drug-related deaths, you can see that the map goes gray, and virtually all data are suppressed because deaths are relatively rare, and Montana is fairly sparsely populated. Worse yet is the fact that we are in 2022 and I'm reporting 2019 estimates, because the most widely used data sources from federal government agencies like CDC and SAMHSA have 2-year lags between when the data are collected and when they're reported. The data are also fallible because people who engage in illicit stigmatized behaviors like substance use are reluctant to report such on a survey. So wastewater data then present this opportunity to obtain more objective measures of drug use in a community. In 2019, Doctors Deborah Kyle and Miranda Margetts at Montana State University led a multi-site wastewater pilot study to look at opioid and illicit drug use in 2 Montana communities, one urban community and one rural. After the data had been collected, they approached Mathematica to help assess the policy value of the wastewater data. And so we analyzed the wastewater data alongside local data that our community partners had provided on law enforcement drug seizures, emergency medical services overdose calls for help, and pharmacy prescriptions. Weekly samples of wastewater were collected over the span of 10 weeks and covered between 1/3 and 1/2 of the entire county's population. The data included information on 18 targeted drug compounds shown here. We compared wastewater-based estimates of use of these drugs with data on drug use from the more traditional data sources shown here, which had much lower population coverage, ranging from 1 person per record to at most 250 people covered per record. You may have noticed that I am not naming the sites included in this pilot study, and that's because, like I mentioned, substance use can be stigmatizing to a community. But masking site names when you present the data, especially on sensitive subjects, is an approach that researchers can use to address any ethical concerns related to privacy or stigmatization. So an obvious starting place for our analysis was to look directly at what's in the wastewater to assess the profiles of drug use between the urban and rural sites. And in doing so, we saw that the most used drugs were the same between the 2 sites, but that methamphetamine use was 6 to 7 times higher in the rural site than in the urban site. Next, we aligned and graphed trends in doses of drugs excreted into the wastewater alongside trends in doses based on filled prescriptions. And looking again at methamphetamines, we saw that the wastewater data, in blue here, suggested much higher use on absolute terms than the pharmacy data. Because prescriptions may not be the main source of meth, which is also manufactured on the black market, it's possible that this analysis is actually capturing a measure of illicit use in the community. That difference between the 2 lines may be that extra black market activity. We then compared wastewater data with logs of drug overdose calls placed to local emergency medical services, superimposing the timing of these calls onto wastewater-based trend lines of drug use. We saw that calls for overdoses involving a particular drug like heroin surfaced soon after high levels of use of that drug in the community. And this pattern held up for many of the drugs that we assessed, and even when most wastewater samples had concentrations below the limit of detection. So by synthesizing these 2 data sources, we saw that the wastewater data might be able to predict when emergency medical services would be called into action. More generally, by bringing together wastewater and administrative data, we can greatly increase the uses and insights that can be gleaned from wastewater. Finally, I wanted to talk about our ongoing work to assess equity in wastewater monitoring. In late 2020, Mathematica led a regional pilot study to assess COVID levels in the rural mountain region of North Carolina using wastewater surveillance. And last fall, we began working with the state to submit the data to CDC's National Wastewater Surveillance System. It's a US federal government program that funds and coordinates wastewater sample collection, analysis, and reporting across the country. And they facilitate peer-to-peer learning among health departments, testing labs, and utilities, all of whom play a very critical role in this work. Recently, we've been assessing whether the wastewater data that the state has been collecting are representative of the larger communities that they're drawn from. Wastewater holds tremendous promise to improve health equity because of broad population coverage, which in the US extends to 71% of households, because of its ability to capture the infections of people with and without symptoms, and because of the ability to register infections among those who may not have the means to visit a doctor. But no work or little work has been done to really say how inclusive the wastewater data are or could be. And so using wastewater data from 19 counties across the state in North Carolina, we're in the process of conducting a geospatial analysis in collaboration with the State Public Health Agency. And the goal of this analysis is twofold. First, we're trying to determine if people connected to sewers that are being monitored through the CDC National Surveillance Program resemble the larger county population that they're drawn from. And then secondly, we're seeing if people connected to any sewer system in a county, monitored or not, differ demographically from those not connected to a sewer system. And as you can see from the maps here, sewer systems can really vary in size from county to county in the US. So for example, on the left, in Durham County, which is one of the most populous counties in the state, 70% of residents are connected to a central sewer system. Whereas in Haywood County, on the right, which is 1/5 the size and which is very rural, only 29% of residents are connected to a sewer, and most of the rest are served by septic systems. So right now, we're in the process of aligning US Census variables on race, ethnicity, education level, insurance coverage, income, employment, and more with the spatial boundaries of the sewer shed so that we can compare metrics like what percent are uninsured who live in the sewer shed versus the percent uninsured in the entire county. And finally, I wanted to describe ongoing work we have with the Rockefeller Foundation to assess equity in global wastewater monitoring programs. This is part of research being conducted in collaboration with the UK Health Security Agency. In early April, we held a global roundtable to discuss ongoing challenges and priorities. And we conducted a follow-up survey of representatives from 43 countries to characterize global diversity and needs around wastewater monitoring efforts. We analyzed patterns separately by low or lower middle income countries, upper middle income countries, and high income countries based on World Bank classifications. India was represented by Dr. Chowdhury and Shirish Harshe, who are leading wastewater surveillance efforts in Bangalore. Based on our analysis of the survey, we're seeing that low or lower middle income countries have had wastewater surveillance in place for less time than high income countries, and they collect wastewater samples less frequently, on average once a week or less, compared to multiple times per week in most high income countries. Whereas all high income countries sampled wastewater from at most 2 locations, a treatment plant and manholes, low or middle income countries often sample from a mix of locations, which points to potential for more logistical challenges in these countries compared to in the US or other high income countries. We're also seeing that fewer low or middle income countries use gold standard approaches for sampling and testing, including composite sampling rather than grab sampling, and digital droplet PCR rather than quantitative PCR. Digital droplet PCR is more sensitive, and so you can detect, one infection out of many thousands of people rather than just a few thousand. These countries also wait longer to get their data from the lab. So the average reporting time was 4 to 5 days in low or middle income countries, which is almost double the reporting time in high income countries. And then finally, whereas all high-income countries had measured viral variants in their wastewater, only 40% of low or lower middle-income countries had done the same, and 55% of upper middle-income countries. And most of these countries were monitoring for variants infrequently. And so taken together, we see that with less frequent and less comprehensive sampling, with sampling and testing based on non-optimized testing methods, and with a lack of routine viral variant monitoring, low or middle income countries may be less equipped to detect that next global threat and less able to protect the health of their populations than high income countries. These data really point to the need for more investment in global wastewater monitoring efforts so that we can create a more robust early warning network. Over the next several weeks, we'll be pulling information from this survey into a commentary that we'll publish. And you can also read more about our work around wastewater data synthesis, triangulation, and translation through other publications shown here. And I'd like to end by thanking TIGS and the many partners who supported and contributing to the work that I highlighted. Thank you for the opportunity to present.
Thank you, Panna, for the very wonderful, very unique presentation, and gives us a lot of food for thought. So I'll now request Dr. Sivaranjani to take the webinar further and a panel discussion. Sivaranjani.
Yeah. Hello. Hi, all. Thank you very much, Dr. Madhvi and Aparna, for the wonderful talks. I am Shivranjani Morir, and I am currently placed at TIGS Virtual Center at CCMB Hyderabad. And I work on wastewater-based epidemiology here in collaboration with CCMB. So I would take up a few questions for Dr. Madhvi and Aparna. I can see that the question answer box has several questions for both of you. And we'll first go ahead with the set of questions that I have, and if time permits, we can take up a few unanswered questions from the question answer section. So the first question is for Dr. Madhvi. It's good to see that you've done extensive work on wastewater-based epidemiology for SARS-CoV-2 in the Indian context, and you were among the first groups to initiate this work in India. So how do you see the use of wastewater-based epidemiology beyond COVID-19? Are you also planning to expand wastewater-based surveillance to other pathogens?
So this is a very good question, Shivaranjani, and I think it fits very well within the topic and the theme of the seminar. So when we talk about wastewater surveillance, certainly, we are planning not exactly the pathogens, but you could say the detection of AMR, that is antimicrobial resistance genes, and how they are transferring. That is one. Another thing is that when we talk about infectious disease monitoring, we have also established methods for detecting some of the vector-borne diseases from the environmental samples, that is dengue and chikungunya, which we are implementing on a pilot stage with the Ahmedabad municipality, so where we are detecting these viruses from the mosquito samples. So these are a couple of things, that we could think of beyond COVID when we talk about environmental surveillance.
Yeah, good to know that. Thanks a lot, Dr. Madhvi. So the next question is for Aparna. Aparna, first of all, thanks for giving the mathematical perspective and a holistic perspective to wastewater-based epidemiology. So my first question is about the sampling parameters and sample source. So we understand that STPs or sewer sheds are the ideal sampling locations for wastewater-based epidemiology, as they are more controlled with respect to catchment area, the number of people contributing, and the volume of wastewater. But in certain scenarios, for example, in Indian rural areas, and few examples that you have even pointed out in your presentation, the sewer system is not well planned. So what sampling or data analysis parameters do you think are important in such scenarios to get maximum and accurate information out of it?
Yeah, it's a good question, and it's something that's even relevant when we go upstream of wastewater plants in the US and go into manholes. And it's, when you have these open drains or surface waters that you're sampling from, you want to try and capture as much of the population that you're trying to reach or trying to monitor as possible. So understanding the schedules of when people are using these drains is important. So if there's a major bus route that goes through an area, and you know that people, get off these buses and they'll use these open drains and then go to work, that's a good time right after that happens to sample from. It's after, substantial people in the community have used a certain site. So for sampling, it's really trying to get a representative sample from the population. For analysis, it's important to know roughly how many people have contributed. So the benefit of these central plants is that it's a very typical population that's connected to the sewer, that's contributing to the treatment plant. But with open drains, it's a very mobile population, so you don't know. So it's important to use, biosensing population like counts. There are a few creative approaches you can use to try and understand how many people have contributed a sample, so that when you analyze and quantify the viral copies per genome per liter, that you can actually normalize that concentration by the number of people who were sampled.
Yeah, so one might not work for the other location. So you actually need to know the parameters relevant to. So thanks. The next question is for Dr. Madhvi. So what is your opinion on the use of wastewater-based surveillance for identification of SARS-CoV-2 variants? You have shown with the example of 7 samples in your study. So especially in the context of emerging variants, how do you see wastewater-based epidemiology?
So this is also very relevant when you are targeting the genome sequencing. Although, your chances of capturing novel mutations at individual cases are negligible. But when you're talking at a community level, which are the key variants which are circulating, if you know their signatures, like in case of Omicron and Delta, they have a specific signature, and I had shown a case incidence of Winzol where even almost a month before, the Delta frequency in the population was around 50.
I think I lost Dr. Madhvi. Can others hear her?
I think
No.
we lost the connection.
Okay, we'll wait for a moment maybe, or else we'll go to the next question then. Dr. Madhvi, we are not able to hear you.
Oh, I am so
Yeah.
sorry. Probably it is because of my connection issues. Now, can you hear me?
Yeah, now we can hear
I
you.
can hear you now, yeah.
I'll just stop my video, probably if it could make my connection better.
Yeah.
So what I was trying to tell here is that we may not be able to capture the individual variants or the novel variants, but at a population level, it is a very important tool to identify, the frequency of some of the variant of concerns, like as I shown in case of Delta, in February first week samples, which was almost 57%. Also in case of Omicron, we could detect it in a very high frequency. So this is really very important. In case of some novel mutation, if it is, spreading in the population at a high frequency, at least we could understand how, the mutation is going to act and behave. So yes, I think it holds a very important potential doing the genomic surveillance.
Yeah, we might not get the complete sequence of the virus, but the signatures can be sufficient to assign, to understand the variants that are already.
So it's a community SARS-CoV-2, and you
Yes.
can see from the wastewater samples also we are able to achieve 60 to 90%
Yeah, 60 to 90%.
uniformity. Yeah.
Yeah. Thanks. So the next question is for Aparna. You've been working on wastewater-based epidemiology for understanding the illicit use of substance or drugs, even way before COVID. So the next question is regarding that. So some drugs might have non-specific metabolites that are similar to other drugs, or in cases where the biomarkers that are associated with the drugs are not very specific. So is there a way to understand their use by wastewater-based epidemiology, or are there any examples of such drugs that have similar metabolites that can?
Yeah, absolutely. So some of the drugs that you ingest are excreted in this parent compound form. So they are identical going in and going out. And then other drugs are metabolized by your body, and what your body excretes is uniquely kind of a signature for that drug. But for something like morphine, for example, morphine, you can measure morphine in wastewater. You can measure codeine in wastewater, but codeine after very little time, so an hour or so, transforms into morphine. And then heroin also transforms into morphine after 1 or 2 hours. So there is a metabolite called 6-MAM, which is unique to heroin, but it's very difficult to measure because it's just unstable in wastewater. So in this way, you have 3 different drugs that all look like morphine in the wastewater.
Okay.
So when you measure morphine, you don't know which of these parent drugs, 2 of which are legal, one of which is illegal, you're actually measuring. But researchers have found a way around this by, again, bringing together many data sources. So if you have prescription records from local hospitals, and you know how many prescriptions for morphine, how many prescriptions for codeine were filled, you can actually parse out those legal uses of the drugs and isolate heroin use, for example, by measuring morphine in the wastewater. So in this way, you're kind of using multiple data sources to triangulate what is legal and what is illegal, but by bringing together many. So that's one example that's been used in Europe and has been used. We attempted to do some of that in Montana as well.
Yeah, so that means one can extract maximum information by complementing the wastewater data along with other data sets as well. So that way it can work the best. Thanks. So the next question is again for Dr. Madhavi. So what are your thoughts on the pan-India wastewater surveillance for SARS-CoV-2? How do you think is the roadmap to do that for a country like India? What challenges do you think we need to overcome to make it work efficiently?
Yeah, I think when we look at a country like India and a very large, population, this is a bit challenging task. And as I mentioned earlier in my talk, that INSACOG is also now trying to establish a wastewater-based epidemiological surveys, at least to begin with, major cities. Because if you look at the pandemic, these have been, the major hotspots like Mumbai, Delhi, and Bangalore, Hyderabad, Pune, Ahmedabad, Kolkata. So these are the regions where, government is trying to establish a systematic surveillance, again, using the pandemic curve monitoring or how, the disease is going. At certain states, I know like Gujarat, which also has a funding agency like Gujarat State Biotechnology Mission. So they have started Pan Gujarat WB surveillance, wherein, along with Ahmedabad and Gandhinagar, they are also funding agencies to survey Surat and Vadodara, which are another larger cities. So certainly, if not at a village level, we can directly go, but we can target the hotspot regions and we can initiate, some systematic surveying programs so that we have a reference data points to compare to, to the previous week data, and that is how we could do monitoring. Along with that, the genome sequencing efforts of these samples also would be very, very relevant to understand what is dominating a particular pandemic at a site. And there are few institutions like IIT Gandhinagar or some of the hospitals who have also started their surveillance programs at their level. But I still strongly feel that if as a country we have a centralized dashboard system, probably through ICMR or maybe DBT, that would be very, very relevant to capture all these wastewater-based epidemiological data for certain meaningful, public health points out of it.
Yeah, thanks. So the next question is for Aparna. What parameters do you think should be considered when you need to extrapolate the wastewater-based data of one small geographical location, like a few sampling sites in a city, to extrapolate to a larger city or town? For example, if we have 10 sampling locations in a city, just averaging them out to understand what is happening at city level might not be the best way to do it. So what all parameters do you think should be considered while doing those kind of analysis?
Yeah, we came across this challenge in North Carolina when we analyzed, the statewide data being collected to see a few counties had 3 different sites being monitored. And we thought, okay, can we just average across those 3 sites and get a more, representative measure? But what you find is that the blips, like the spikes in one area, will get flattened because you're averaging with other areas that don't have that spike. So you just end up with kind of a line that's not very informative. So what we found is it's important to monitor these sites separately and to normalize and to do all of your analysis separately, but to look at them together, not just within the site, but regionally. And with an infectious disease, one case can become 10, can become 100 so quickly. And so what you want to know is, yes, no, is it present in any of the 10 sites, say, being monitored? And if so, then maybe flag, the alarm. So taking a kind of a conservative approach to say, if this virus is present in any of these 10 locations, or if it's, the trend is starting to rise in any of those 10 locations, then you may warn the officials that, to look carefully. And then second order is to say, okay, which of these 10 locations are we seeing this? And is there kind of a regional spread that you can see in the wastewater data? So I think the first is just to say, look at them separately, but develop your metrics for warning and risk scores based on taking all 10 sites data together.
Thanks a lot. So with this, I thank our today's speakers, Dr. Madhvi Joshi and Aparna Keshriya, for their insights on wastewater-based epidemiology. I'm sure they have convinced all of us that wastewater-based epidemiology is a cost-effective and unbiased approach that gives a widespread and objective picture of the real situation. So once again, I thank Dr. Madhvi and Aparna for being with us. And I thank Dr. Rakesh Mishra for the welcome address and introduction to the topic and the speakers. And we would end this session by thanking the audience for patiently listening to the talks and their questions. Thank you all. Thanks.
Thank you.